Okay, imagine a future, maybe not so far off, where we could gently guide our body's own repair mechanisms, nudge them back toward their prime.
Mm-hmm.
Today, we're doing a deep dive into a really exciting area of science. It's all about rejuvenating our regenerative cells, basically helping them work better for longer.
Right.
We're looking at a pretty groundbreaking article called Niosomes, A Gentle Path to Stem Cell Rejuvenation. It introduces a truly innovative strategy.
It really does.
This
And the real aha moment here, I think, isn't just about a new system. It's more a strategic shift in how we even approach cellular rejuvenation.
a strategic shift. How so?
Well, for a long time, gene delivery methods were complicated. You had issues of permanence, immune reactions, all sorts of hurdles.
Yeah, I remember the viral vector concerns.
Exactly. So this deep dive should give you a bit of a shortcut to understanding how Niosomes, with their, let's call it, gentle, transient approach, could really reshape our thinking. It's less about rewriting the code permanently, and maybe more about giving nature an intelligent, temporary boost. Our mission today is really to understand this promising, non-invasive way to enhance aging mesenchymal stem cells, MSCs, without that permanent genetic alteration.
Got it. Okay, so for our deep dive today, let's zero in on those mesenchymal stem cells, the MSCs. Many of you listening will know these are incredibly versatile repair cells, often called our body's internal maintenance crew, right?
That's a good way to put it. Yeah, they're typically found in places like bone marrow or maybe umbilical cord tissue.
And they're crucial for tissue repair regeneration.
Absolutely crucial. But, and here's the catch, like all our cells, MSCs aren't immune to time. They age. They experience what's called cellular senescence.
Senescence. Great.
Think of it like this. These repair cells, they get old, they get tired, they start to lose their ability to divide efficiently and, effectively mend tissue.
Okay, so they stop working as well.
It's worse than that, unfortunately. What's truly problematic is that these senescent MSCs don't just become inactive bystanders. They develop this, particularly nasty habit called the senescence-associated secretory phenotype, or SASP.
SASP. Okay, what does that mean?
It means they start actively secreting a kind of nasty cocktail of inflammatory molecules, things like cytokines, chemokines, enzymes that actually degrade tissue.
Wow.
So not only do they stop doing the repair job, they actually start contributing to inflammation and damage. They can potentially drive age-related diseases throughout the body.
So your internal maintenance team isn't just, taking a coffee break. They're actively spilling oil and leaving rusty tools everywhere. That is not ideal.
Not ideal at all.
And this is precisely where the researchers stepped in with this clever idea involving niosomes.
Exactly. Niosomes are, they're fascinating. Think of them as incredibly tiny, almost like intelligent delivery vehicles. They're nano-sized, non-viral systems specifically designed to carry genetic material.
Nano-sized.
Specifically, they're little bubbles, you could say, nanovesicles, made from a combination of non-ionic surfactants, basically a type of lipid or fat and cholesterol.
Cholesterol, interesting.
Yeah, and this unique composition gives them a significant edge over older methods, like those viral vectors we mentioned. For one, they seem to have very low toxicity.
Ah, so less likely to provoke an immune response.
Much less likely, yes. That's a big plus. And the cholesterol isn't just filler. It actually gives them remarkable stability and rigidity. That's crucial for them to stay intact as they navigate the body.
Okay, stability. Makes sense.
And from a practical angle, their formulation can actually be adapted to meet good manufacturing practice, GMP standards.
Right, GMP. That's huge if you ever want to get this into clinics.
It's absolutely essential. Now, how they work? Once they reach a target cell, the cell basically swallows them whole. It's a process called endocytosis. Then once inside, these clever niosomes manage to escape from the cell's internal packaging bubbles, the endosomes, and release their genetic payload directly into the cell's main workspace, the cytoplasm.
And that's where the genes can start working.
Exactly. That's where they get to work.
So it sounds like we've found potentially a much gentler, maybe more efficient way to get these beneficial genes into our cells without all the traditional baggage and risks.
That's the hope.
But what exactly are these niosomes delivering? What's the payload that's designed to give these sort of tired, aging cells a boost? And why these specific components?
Great question. The what and the why. The researchers chose two genes specifically known to support cellular repair and function. First, there's BMP-7. That stands for bone morphogenetic protein 7.
BMP-7.
It's a signaling molecule crucial for bone formation, yes, but it's also a broader player in tissue regeneration generally.
Right.
The second one is GAP43, that's growth-associated protein 43. It's pretty well known for its role in neuronal growth and cellular plasticity, essentially how adaptable cells are.
Okay, so BMP-7 and GAP43, supporting regeneration and adaptability.
Precisely. But here's the really crucial part, and it's a massive safety and strategic advantage. These genes were expressed transiently.
Transient, meaning temporary.
Exactly, temporary. The changes they produced in the cells didn't last forever. They didn't permanently alter the cell's genetic blueprint, its genome.
Ah, okay. That feels important.
It's incredibly important. It's not just safer in the immediate sense. It opens up possibilities for, say, repeated treatments or adjustable therapies that permanent changes just can't offer. You could potentially fine-tune a treatment over time without making irreversible alterations. That's a really powerful concept in regenerative medicine.
That transient expression really does sound like a potential game changer. It allows for that flexibility. So, okay, you've got this clever delivery system, these targeted genes. What did the initial lab results actually show? Did these tiny bubbles manage to, rejuvenate those tired stem cells?
Well, the in vitro results, so experiments done in the lab, cells in dishes, they were indeed very encouraging. The MSCs that were treated with the niosome-delivered BMP-7 and GAP43, they showed a clear increase in proliferation.
Proliferation. So more dividing, more growing.
Exactly. They were dividing and growing much more robustly, which is precisely what you want from your regenerative cells.
Okay, good start.
And maybe even more telling, there was a noticeable reduction in markers associated with senescence. Things like senescence-associated galactosidase.
The aging markers went down.
They did, which suggests a genuine reversal of at least some of those aging characteristics we talked about earlier. And critically, these cells didn't just multiply, they maintained their vital ability to differentiate into other cell types.
Oh, so they could still specialize. They kept their function.
Yes, exactly. It indicates their core functional capacity was preserved and maybe even enhanced. Now, the paper didn't provide really specific quantitative details like exact fold changes or percentage improvements.
Okay.
But the biological signals observed were overwhelmingly positive, certainly for this early stage of research.
It's genuinely exciting to see those positive signs in the lab, especially, like you said, retaining that differentiation capacity. But, and there's always a but, these are in vitro results, lab dishes.
Right.
And while this sounds incredibly promising, the history of regenerative medicine is, well, it's full of initial breakthroughs that then face pretty significant roadblocks. What are some of the biggest real-world challenges that this niosome approach has to overcome before it can even think about moving from that petri dish to, say, a patient?
You've absolutely hit on the most critical phase, that leap from in vitro to in vivo, from the lab dish to a living organism, and then eventually to clinical human trials. It's monumental. It really is.
Yeah.
One primary concern is immune interaction. We don't yet fully know how the human body's immune system will react to repeated or maybe large-scale niosome administration.
Right.
Will it eventually see them as foreign invaders?
Right. The body's defense is kicking in.
Could happen. Then there's the very practical challenge of biodistribution and stability.
Meaning getting them to the right place and making sure they last.
Precisely. Can these niosomes reliably reach the specific tissues or cells we're targeting within the incredibly complex environment of the body? And will they remain stable enough in circulation to actually deliver their payload effectively when they get there?
Lots of variables there.
Huge variables. And long-term safety, of course, is also a massive consideration, particularly if repeated treatments turn out to be necessary. We need to rigorously assess the risk of any unintended cellular stress or, off-target effects. And naturally, the regulatory hurdles are immense. Agencies like the FDA or EMA demand incredibly robust evidence of safety, efficacy, and perhaps most importantly, reproducibility before anything gets near a clinic.
Those are definitely very tangible challenges. But thinking beyond those practical hurdles, does this research give us any clues yet about whether we're truly reversing aging at some fundamental level here? Or is it more like we're just making the cells act younger for a while? That feels like a really profound distinction to make.
That's a really powerful question. And it gets right to the heart of what anti-aging truly means, doesn't it?
Yeah.
Right now, this research suggests a powerful functional improvement. The cells work better. But to determine if it's a fundamental reversal versus just a compensation, honestly, we need much more mechanistic insight.
Meaning understanding the how.
Exactly. Understanding precisely how these delivered genes are influencing the complex cellular pathways involved in aging and regeneration. That detailed understanding will be absolutely essential to fully grasp its impact and its true long-term potential.
That's a vital point for the future of this whole field, I suppose. But looking at the core of what we've discussed today, this research really does stand out. It feels like a thoughtful and technically sound advance.
I agree.
It represents maybe a significant refinement in how we pursue regenerative therapies by enabling this transient, targeted gene delivery without that permanent genetic modification. It feels like a solid step in the right direction for precision medicine.
Absolutely. And
Right.
while, we have to stress, these findings are preclinical,
Right, very early stage.
very early.
Yeah.
We're not talking about an imminent cure or immediate clinical application, but for anyone tracking the frontiers of anti-aging biotechnology and regenerative medicine, this is definitely more than just another promising study. It represents a really significant shift in approach, I think, and it's definitely worth watching closely.
Well said. And that leads us to a fascinating final thought for you, the listener. Given this gentle, transient approach with Niosomes, where the goal isn't necessarily to permanently rewrite the body's code, but maybe just to give it a temporary, intelligent nudge back towards youthfulness, how might interventions like this change our fundamental perspective on tackling age-related decline? And maybe think about what new, perhaps even unexpected possibilities might this open up for enhancing human health and longevity in the decades to come, perhaps beyond what we've even touched on today.